let's give a quick revision to polymerase chain reaction and blotting techniques. So first of all, what is PCR? PCR is basically a technique in which under laboratory settings we can amplify DNA from any source, whether it is from virus, bacteria, plant or animal, we can amplify it. So if we can amplify DNA, it is easier for us to diagnose and for research. We can diagnose any abnormality in DNA with help of this technique because DNA is amplified many-fold. So what is the process? So in it we use enzyme DNA polymerase to multiply the DNA. And with each cycle DNA is doubled. So with each consequent cycle the DNA is doubled. So there is exponential amplification. And the amount the DNA which is amplified we can separate it with help of electrophoresis and detect with help of certain blotting techniques. So the PCR, with help of PCR, we can amplify DNA from a single cell, from a single hair, from a small drop of blood or semen. So it is easier to amplify. So because we can amplify, we can then find the source also. So each cycle doubles of the copy number of the amplified gene. So like for first there is 2, then it will become 4, 8, 16 and so on. So within few cycles we can multiply DNA to its billion-fold. So here you can see this is a gene of interest. So in this gene there are 2 copies which under first cycle become 4, then 8, then 16, then 32. So in this way there is exponential amplification and within few hours we get a billion-fold of the DNA of interest. So what is required for this procedure? First of all we need DNA of interest. Then we need deoxyribonucleotides. We need primer. That is the primer which is complementary to flanking region of DNA of interest. That is the DNA which is the sequence of DNA which is just before the DNA of interest. So we should know the base sequence of the DNA which is just before the DNA of interest. There is no need to know the base sequence of DNA of interest. Because we need a primer, we need to construct a primer for the flanking region. Then we need a polymerase enzyme. It should be thermostable because in polymerase chain reaction we need enzyme which could be stable at high temperature, that is 95 degrees centigrade. So this thermostable polymerase is Taq polymerase which is isolated from Thermus aquaticus, which is a heat stable and does not get denatured at high temperature. So these are the things which we require: DNA template, primer, polymerase enzyme, nucleotides and certain buffers and cofactors. And this is a machine, PCR machine, which is also known as thermal cycler. It helps us to amplify the DNA of interest. So the procedure is, there are 3 steps which are repeated again and again in every cycle to double the amount of DNA. So first step is denaturation. For denaturation, temperature is very high, 96 degrees centigrade. DNA become denatured so that it could separate into single strands. Then is annealing. Then we want to anneal the primer to the DNA. So we lower the temperature to 50 degrees centigrade. So the primers anneal with the single-stranded DNA. Then is extension. So at this temperature, that is 72 degrees centigrade, the enzyme polymerase could synthesize new DNA. So the enzyme Taq polymerase synthesize new strands of complementary DNA. So this is how there are 3 steps. First of all, there is denaturation, then annealing of primers, and third is extension. So at end of these steps, 2 copies of DNA are formed or the DNA amount is doubled. Now, why we need PCR? Why we need to amplify DNA? It is required for clinical diagnosis of certain diseases. It is required for detection of HIV at very earlier stage because HIV has very long latency period. So it is not possible to detect HIV at earlier stage. But if we amplify the DNA, we can easily detect with help of PCR. Then we can detect cancer, bacterial infections like tuberculosis in very earlier stage, even when the DNA amount of these bacteria or cancer is very low. Then for prenatal diagnosis, DNA from chronic villus samples or from the cells for amniocentesis are amplified by PCR, and it helps in prenatal diagnosis of sickle cell anemia, beta thalassemia, cystic fibrosis, etc. Then we can compare normal genes with mutated gene with help of PCR, because it will amplify the DNA to many fold and it's easier to compare between two. What is the difference? Then for forensic analysis, so with help of PCR, we can isolate DNA isolated from single human hair. A tiny spot of blood or semen can be amplified so much that we can easily detect its source, from which specific individual it belongs to. Then it's also helpful in archaeology and paleontology. It is amplification of rare fragment of DNA which are obtained from extinct species could be amplified and then compared with the living species. This will help us to reveal the missing links in evolution. So it is also helpful in studying about archaeology and paleontology. Then one thing more you should know is the role of PCR in COVID detection. So we use PCR technology to detect the COVID cases. So if the viral load of COVID virus is less, then what will happen? We need many cycles of PCR to amplify and detect it. But if viral load is more, we need lesser cycles of the PCR to detect the virus. So according to ICMR Guidelines, if the virus is detectable in 35 cycles or earlier, the patient is considered positive for COVID. So the CT value is important for diagnosis of COVID, because if the viral load is more, then in lesser cycles the DNA will be amplified so much that we can easily detect the DNA. Then the various types of PCR reaction. The first one, which is simple PCR, which we have already discussed. Apart from this, there are other PCR techniques like nested PCR, quantitative or real-time PCR, reverse transcriptase PCR, and multiplex PCR. So the second one is nested PCR. This type of PCR is used when DNA of interest is in very small amount as compared to the total DNA in the sample. So by this process, we can amplify the DNA of interest. So first we will use outer primers to amplify the whole DNA. Then we will use inner primers to amplify the DNA of interest, so that we can have DNA of interest in sufficient amount. So this is how there are outer primers and inner primers. So we use two types of primers in nested PCR. Next is quantitative or real-time PCR. So what is the difference between this and simple PCR is that we can quantitate the amount of DNA simultaneously with the amplification. So how can we quantitate? We can use certain dyes like ethidium bromide or cyber green, with help of which we can quantitate simultaneously with amplification. And second process is by using probes, which are complementary to DNA of interest. These probes generate fluorescence when they hybridize with the DNA of interest. And because of these fluorescence, with help of scanners, we can quantitate them. The various probes used are Tachman, molecular beacon, and fluorescence resonance energy transfer probes. Next is reverse transcriptase PCR. In this type of PCR, first complementary DNA synthesized from mRNA, and then this complementary DNA is amplified and then it is used to synthesize useful proteins. So because it is first DNA is formed from mRNA, it is known as reverse transcriptase PCR. Next is multiplex PCR. In this, we can simultaneously multiply many targets using different primer in one reaction only. So in one reaction, we can multiply different sets of DNA by using different primers. So this type of PCR is known as multiplex PCR. Next are blotting techniques. Now we have amplified DNA, but how can we detect? whether this is DNA of interest or whether this DNA belongs to certain disease or this is a mutant DNA, we can detect with help of blotting techniques. So there are different blotting techniques with which we can identify specific DNA, RNA or proteins from thousands of contaminating non-target molecules in complex samples. So for DNA, we use southern blotting. For RNA, we use northern blotting. For proteins, we use western blotting. And to study about protein-protein interactions or protein nucleic acid interactions, we use southwestern or overlay blot. So this you should know different types of blotting techniques which are used for different interest, whether it is DNA, RNA, protein or interactions. So first of all is southern blot, which is used in DNA to identify the DNA of interest. So the process is first the DNA of interest from the cell is cut into smaller fragments with help of restriction endonuclease enzyme to become the restriction digest, and these different lengths of DNA is subjected to electrophoresis. Now as DNA is negatively charged, it will move towards anode, but the smaller fragments of DNA will move faster and further. So with help of this we can separate the different DNAs according to their size. Then we will transfer this to the alkali solution so that the DNA could be denatured and could be converted to single strand. Why it is important? Because the probes which we use to identify can hybridize only with the single stranded DNA. So once it is denatured, it is then transferred to nitrocellulose membrane. Why it is transferred to nitrocellulose membrane? Because in gel state it can leach out. So to fix it, we transfer this to nitrocellulose membrane. After that, we subject it to various radioactive probes and under the suitable temperature, and after certain time, these probes can hybridize with DNA of interest and then emit, because they are radioactive or they are fluorescent, they can be easily detected with help of autoradiography or fluorescent scanners. So in this way, we can detect the DNA of interest with help of blotting technique. So this was for DNA. Then another blotting is for RNA. So we use radioactive labeled complementary DNA probes. For Western blotting, which is for detection of proteins, we use antibody probes. And for Southwestern and overlay blot, they are used for understanding or for studying the interactions between protein and nucleic acid or protein-protein interactions. So again, these are three types of blotting: for blotting of DNA, for detection of DNA, RNA or proteins. And there are different probes for each one. Then there is dot blot technique, in which samples are directly applied to the filter and not subjected to electrophoresis. And they are directly applied to filters and then they are subjected to probes to detect. They are not undergo electrophoresis, but this process is lesser sensitive. So that was all about polymerase chain reaction and blotting techniques. So it is a very important and useful process which is used to amplify the DNA of interest. It helps us in diagnosis as well as in research process. So you should know about the procedure of polymerase chain reaction, the cycles, and with each cycle how the DNA is amplified. There is exponential growth. Then you should know that we need the polymerase enzyme from Thermus aquaticus because it is thermostable. Then we should know about the various blotting techniques. Southern blotting is for DNA, northern is for RNA, and western blot for proteins. So that was all. Thank you so much.